Very pleased to be here speaking to you today about the Australian Dementia Network trial screening activities. So Perminder's already filled you in on my first slide, which was just to mention what the Australian Dementia Network is, and it has three principal components: the registry, the clinics and trials. It has approximately 15 institutions affiliated with it, and that number continues to grow. You've heard that there's the Clinical Quality Registry, which is for newly diagnosed dementia and mild cognitive impairment, with the aim to try and recruit as large a proportion of these people from across Australia as we can, and then follow them over time, basically from diagnosis to death, to monitor the quality of their care and to use that to feed back where there are deficiencies to caregivers, government authorities, etc., to generally improve the level of care. It's also looking like it's going to be a very good mechanism for assessing post-marketing surveillance of the new disease-modifying antibodies that appear to be coming to Australia, or likely to be coming to Australia within the next 12 to 18 months. The memory clinics you've heard about, the idea is to get them together to standardise and support and expand the function of memory clinics and introduce new diagnostic techniques. And the third part, which I'm talking about, is the national trial screening for finding individuals with preclinical, prodromal and mild Alzheimer's disease and potentially other types of dementia for trials of various types, not only pharmaceutical, but also lifestyle modification studies. So ADNeT trial screening, it's a fairly simple concept. It's been running in Melbourne since 2014. And we in Melbourne are getting about 300 referrals of patients with mild cognitive impairment or early Alzheimer's disease to the coordinator. There's a phone screen and then some on-site screening. And provided they meet general criteria for clinical trials, they're open to the idea of going into a clinical trial, they will then get an amyloid scan and a tau scan, an MRI and a detailed cognitive assessment. After that, they end up being referred to a trial cohort or longitudinal study or put directly into a trial. But that decision is made in conjunction with the referring doctor and the patient. So we tailor the trial to the patient. This is one advantage of taking this particular route rather than going directly to a particular clinical trial. The program is now running in Perth and is just about set to kick off in Brisbane and Adelaide and Sydney. The referral is quite simple. We have this simple referral form on the right. This is the one we use in Melbourne, but you can use it for all of Australia at the present time. Just put the name of the referring specialist, check the eligibility criteria, which is probable MCI or mild AD, aged 50 to 85, availability of a reliable study informant or partner, formal education of at least 7 years, fluent in English, mini mental state score of greater than 20, capable of tolerating neuropsychological assessment, and no serious unstable comorbid conditions. So if they fill that, you then tick if they've had any investigations of these, well, if they've had an MR and FDG PET in the last 12 months, and hopefully you'll attach a letter to give us a bit more detail, and then also indicate whether you want the feedback of the results to come from you or from the ADNeT trial clinician. Each ADNeT screening site has a specialist memory clinician, either a geriatrician, psychiatrist or neurologist, who provides feedback to patients. So you send that off, email it, and then we get in touch with your patient and follow through. There is also a portal for normal volunteers to register their interest. So this is how we are attempting to identify people with preclinical Alzheimer's disease. In other words, they've got amyloid plaque, but they're cognitively normal. So you can access this portal, well, you will be very shortly able to access it. It's not there right this minute, but we expect to have it up within 2 weeks, by going to the Australian Dementia Network website. It's all one big long word, www.australiadementianetwork.org.au. This website will collect details on contact details, medical details, including existing diagnoses. If they happen to be given a diagnosis of MCI or dementia, they will expect most of the people to go through this portal to be healthy elderly people. We get information about their level of subjective cognitive complaint, particularly looking for high risk features, such as if others have mentioned to them that they're concerned about their memory, or they're convinced there's been a significant decline in the last 3 years. We elicit a history of first degree family history of dementia, and we then send out spit kit tests for ApoE4 testing to those people aged between 55 and 75 who look like they're going to prove suitable for trials. They're then brought in, not everybody, but we select based on high risk characteristics in the first instance, and these are being an ApoE4 carrier, having a high risk subjective memory profile, complaint profile, having first degree family history of dementia with onset before the age of 75. We bring them in, do some in-person assessment, and then basically put them through the same program as we used for the MCI-AD patients. So amyloid PET or a CSF if amyloid PET's not available, MRI scan, blood collection, full cognitive battery, plus or minus a tau scan. After this, again, we have a expert talking to the patient, discussing potential clinical trials. Now, there is one coming to Australia for an anti-amyloid therapy. It's called the AHEAD study or the A345 study. This is funded a joint public-private partnership through the NIH in the United States and Eisai, the pharmaceutical company. They are supporting our trial screening activities to help find patients for this study. There's another study, a lifestyle modification study called ARROW, which is an academic study, which in Australia is being led by Ralph Martins, who the patients that we find through this screening mechanism may also be suitable for. But we're open to approaches about supplying subjects for other studies too. Now, to set up this network and to screen people optimally, particularly for the AHEAD study, this A345 study of preclinical AD, where the traces to be used in that are NAV4694 for amyloid imaging, because it is the most sensitive tracer, and MK6240 for tau imaging, again, because this is the most sensitive tracer for tau. We have established and almost have operational 5 sites throughout Australia. Melbourne site has been up and running for several years. Perth site has just come online with both. They've been making NAV for quite some time, but they've just come online with producing MK. And we have Adelaide, Sydney and Brisbane sites also who will be online by the end of this year producing these traces. And these tracer production sites work in conjunction with our screening centres. At the moment, we have established a centre in Melbourne at the Florey Institute. It's a composite centre, Florey Institute, University of Melbourne, Austin Health, and inquiries or referrals go to Lucy McIntosh. At Macquarie University, we have a site, and that is the contact person there is Cynthia. University of Newcastle is under development at the present time. They will be only looking for normal volunteers for the preclinical study, and Renata is the coordinator there. At Edith Cowan University, collaboration with Australian Alzheimer Research Foundation, Mark Rodriguez is their coordinator, with Kevin Taddyke supplying backup supervision. At SAMRI in the Royal Adelaide, we have Alexandra Wade as the coordinator, and the University of Queensland Royal Brisbane Hospital we have Elizabeth Arnold. So at the moment we can take referrals in most sites, although as I mentioned, there has been some delay in getting access to the required traces in Adelaide and Sydney and Brisbane, but that is being resolved. The COVID crisis, unfortunately, has made transport of traces interstate somewhat difficult, but if you have suitable subjects, you can refer them right now and just send the referral to adnet-screening@unimelb.edu.au, and they will be distributed to the appropriate state-based coordinator for follow-up. I'll show this slide again at the end in case you want to write down further details. But remember, adnet-screening@unimelb.edu.au. ADNeT already does have a feather in its cap. It has brought the Clarity study from Eisai to Australia. So this is a phase 3 study of an anti-amyloid antibody. And I think Michael Woodward will talk about this some more. But Eisai were not going to bring this study to Australia, but when they heard about the ADNeT setup and network and the potential to speed the recruitment for their trial, they changed their mind and they have now initiated trial sites in each of the major mainland cities. So this is all very well. It helps us find people for trials, and that in turn will reduce what is really quite a prolonged recruitment phase for most clinical trials. It'll make the trial sites bigger, better, and more viable in the long term, improve their sustainability. But this approach also helps the clinician and the patient by making available latest technology investigations that otherwise are inaccessible. And it does this free of charge. So here's the diagnostic pathway for a patient with a cognitive problem: family history, patient history, assessment, cognitive assessment, some structural imaging, and then access to biomarkers is desirable. So this is what we're talking about. By referring suitable patients to the ADNeT trial screening, you get access to the latest biomarkers. Why do we need biomarkers? Well, correct diagnosis and correctly predicting the future of a patient presenting with cognitive impairment or dementia is hard. The clinical criteria currently used in Australia haven't changed since 1984. And they only have sensitivity between 70 to 80% and specificity of 70% for Alzheimer's disease compared to neuropathological examination. And this is despite the diagnosis being made in top centres. So this paper came out in Neurology in 2001, 13 post-mortem studies showed only about 70 to 80 and 70% sensitivity and specificity. A paper from the National Institute of Aging AD centres in the United States, so centres specifically funded for AD care and research, found similar results: 70% sensitivity, 70% specificity. So it's no exaggeration to say that 30% of people who are given a diagnosis of Alzheimer's disease don't actually have it. This has been seen time and again when people have been scanned with amyloid PET because they've been referred to a treatment trial, supposedly because they have Alzheimer's disease. But in fact, 30% of referrals in general are negative and therefore do not have Alzheimer's disease. The other problem with the clinical criteria is officially you're not supposed to make the diagnosis until the patient has dementia. And patients are presenting earlier and earlier and don't want to wait around 3 to 5 years when they have a significant cognitive problem and they've been told, well, you've got mild cognitive impairment, we'll just have to wait and see if it turns out to be Alzheimer's disease. So mild cognitive impairment is a symptom, it's not a diagnosis, which is a trap a lot of people fall into. And in fact, only 50% of people that meet criteria for mild cognitive impairment will progress to Alzheimer's dementia. About 15% or so will develop other dementias, But importantly, 30 to 40% of people with MCI never develop dementia. And there's no way to sort these out unless you can employ biomarkers like amyloid PET and CSF. So structural imaging, neuroimaging, we've got CT scanning. We look at that to see if there's atrophy in the medial temporal lobes, and with the newer generation CT scanners, you can actually get a fairly good idea of this. So a cognitively normal brain at the top, followed by a clearly atrophic, severe hippocampal atrophy brain and Alzheimer's disease. MRI, we like to use the coronal sections back at about the level of the mammillary bodies here, and look at the volume and particularly the CSF space around the hippocampus. So this is quite normal, whereas this person has atrophy of the hippocampi and increased CSF space, and that is probably a grade 2 to grade 3 example of hippocampal atrophy. FDG PET is available in some centres, and we actually have a submission before the Medicare Services Advisory Committee at the moment to try and get FDG PET reimbursed as a substitute for brain SPECT. And that's looking quite promising. And this is the typical pattern you find in FDG PET and Alzheimer's disease, which is bilateral parieto-temporal hypometabolism. Then amyloid PET, here is a negative scan showing some mild non-specific binding to white matter. And here's a positive scan where you can see the binding around the cortex because there's amyloid plaque there for the tracer to bind to. So FDG PET shouldn't be underrated. It's very good to assist the diagnosis of Alzheimer's disease, but is also good for looking at other dementias. So dementia with Lewy bodies, not only do you have parietal reduction, but you have occipital reduction, both medial and lateral. In behavioural subtype of frontotemporal dementia, frontal lobe reduction, some reduction here along the insula, and in the medial regions, anterior cingulate and anterior caudate nucleus. Progressive non-fluent aphasia, you get it in the left hemisphere in the premotor frontal cortex or dorsal frontal regions, affecting Broca's area, and you can see it here lateral and medial. And semantic dementia subtype of FTD, it affects the anterior temporal lobe. This is a display that quantifies an individual's scan compared to a normal database, and I actually insist upon it when reporting my FDG PET scans. But FDG does have limitations. The degree of hypometabolism does reflect the clinical deficit severity, so findings actually are quite subtle in MCI. So it doesn't have particularly high sensitivity for AD in the MCI stage. Changes also become less clear in the more elderly. So in patients over the age of 75, FDG PET becomes harder to read. And overall, the sensitivity is probably only in the area of about 85%, specificity 70 to 80%. So there's room for improvement. So back in 2002, the first human being had a beta amyloid PET scan with PIB. This is showing it. It was developed from the University of Pittsburgh, and the red areas are showing where this tracer is bound to amyloid plaques in the cortex. It was pretty obvious early on that this was a great test for distinguishing frontotemporal dementia from Alzheimer's disease. No plaques in frontotemporal dementia, so no tracer binding. Not so good for dementia with Lewy bodies. The pattern of uptake is similar, as 50% of patients with DLB have amyloid plaque. That was a carbon-11 tracer. F18 traces were subsequently developed by commercial companies and validated against post-mortem. They had to be compared, the visual read of positive or negative had to be compared with the CIRAD criteria, which meant there were at least moderate or there were frequent neuritic plaques present. And it turned out that all the traces worked very well with sensitivity and specificity averaging over 90%. So here we have a negative scan on the left, again showing just a white matter pattern. Positive scan on the right. We've lost that white matter pattern because the cortex is now filled in with plaque, so the tracer is now binding in the cortex. And it's recommended that you read these scans, most of them in black on white, by looking at this gap between the white matter binding and the edge of the brain. In here, in this negative study, there's no binding because there's no plaque. Whereas in this patient, you can see it's filled in right to the edge of the brain because there's amyloid there. Not all PET traces are the same though. Here's why we use NAV4694 in ADNeT. It is a superior tracer. This is the same patient and the colour scale has been set to the same level of activity in the brain, and yet you can see much more clearly uptake in the cortex, caudate nuclei, consistent with the known distribution of amyloid plaques than with this other F18 tracer. In 2014, the International Work Group set up new criteria for Alzheimer's disease. They dropped the need for dementia. They do require objective episodic memory impairment and a history of progressive disease, progressive decline, plus evidence of a pathophysiological AD process. So that can either be a positive amyloid PET scan or CSF with a low A-beta 42 and high tau. FDG and MRI, FDG PET and MRI, were then relegated to being used for assessing disease severity and monitoring progression if required. Now, there is a catch to amyloid imaging. Amyloid builds up incredibly slowly, over about 30 years before it gets to the level typically seen in patients with mild Alzheimer's disease. And we know that the incidence of Alzheimer's disease doubles every 5 years. So not surprisingly, the prevalence of a positive amyloid scan is quite high with advanced age. And this is what we found in the ABLE study. It goes from 5% of people in their 50s to 10% in their 60s, 30% in their 70s. And if you're 80 to 85, you have a 50% chance of having a positive amyloid scan. So a positive amyloid scan could be a coincidental finding in someone who has cognitive problems for another reason. So it alone cannot confirm Alzheimer's disease. Certainly the absence of amyloid does reliably exclude Alzheimer's disease. But having a positive scan, particularly when the brain is absolutely full of amyloid, particularly in the younger patients, is strong confirmatory evidence for Alzheimer's disease. There are programs emerging designed for clinical practice that will do an automated analysis and give you quantitative results that can help you in your clinical practice. There is a new scale that's been developed called the Centerloid scale, where the amyloid scan can be measured and the amount of amyloid can be quantified between 0 and 100. 0 being no amyloid, and the appearance of the scan is what you get in normal young people, and 100 being the typical amount of amyloid present in mild Alzheimer's disease. We've now got some neuropathological correlation with these Centerloid results and also some prognostic information from these Centerloid results. And basically the cutoff is about 25 Centerloids for a positive scan if you're trying to meet those CRAD criteria for moderate or negative. There's no plaques if the scan has a less than 15 Centerloid. A few scattered sparse plaques or some diffuse plaques if you're between 15 and 25. Then when you get over 25, tau doesn't start appearing in the brain. The tangles don't start appearing till you get, this is in the neocortex, till you get to about 40 to 50 Centerloids. And you have to have about 50 Centerloids before at post-mortem you would find that the Alzheimer's disease neuropathological criteria are met. So this requires the presence of tau tangles as well as amyloid plaques. And as I said, 100 is the typical average result in patients with mild to moderate Alzheimer's disease. Now, in a patient with mild cognitive impairment, if you have less than 25 Centerloids, which is the top two rows here, the grey and the green, there is no progression to dementia over 4 years. If you have a brain with a fair bit of amyloid in it though, over 50 centerloids, which is the red and the orange, you can see you decline quickly and you have a 2-year risk of 50% of having developed dementia. If you have a moderate amount of amyloid present, so 25 to 50 centerloids, it takes about 4 years before you have a 50% risk of dementia. So the amount of amyloid does have prognostic implications. In fact, you can see here, if you have less than 25 centerloids of amyloid in your brain, your memory performance actually improves over time due to practice effects. There has been a large study in the United States to look at the management impact of amyloid PET. It was called the IDEAS study. They found pretty stunning results. Change in Alzheimer's disease drug use versus if the scan was positive or the scan was negative, you can see that it went up by 1/3 in people with a positive scan, went down by about 10% in people with a negative scan. As suggested, the pre-PET diagnosis for Alzheimer's disease was pretty terrible. Only 61% of them had a positive amyloid PET scan. The pre-PET diagnosis for a non-Alzheimer's dementia was equally as bad, with half of them actually having a positive PET scan. So the amyloid PET led to substantial changes in diagnosis. With a positive scan, the rate of AD diagnosis increased from 78 to 95%. But look at what happened when the scan was negative. The rate of AD diagnosis decreased from 73% down to 14%. So huge impact on diagnosis. Now, CSF is also a very good way to diagnose Alzheimer's disease. It's sensitive and it picks up the changes early. What you look for is a reduction in Aβ42. It's thought that this particular peptide of amyloid gets trapped in the plaques, and therefore there's a reduction in the CSF. And you look for an increase in phospho-tau, up about 200% in your typical Alzheimer's patient. This phosphorylated form of tau is abnormal and relatively specific for Alzheimer's disease. Total tau also goes up, but that tends to be a general reflection of brain damage, and so can go up in other neurological disorders. There's a good concordance between an abnormal CSF result and an abnormal PET result. As you can see down in this quadrant here, this is high amyloid and low Aβ42, remembering that Aβ42 goes down in Alzheimer's disease in the CSF. And here in the negative quadrant, you can see most patients are actually in the negative or the positive quadrant, and only a few that are discrepant. So we've had Alzheimer's amyloid imaging for some time. Tau imaging has been relatively recent. These are some examples that we have in patients who are amyloid positive with a clinical diagnosis of Alzheimer's disease. Interestingly, there are 2 patients here who had negative tau scans. When we looked closer at this particular case, we found bilateral posterior cerebellar infarcts and a small occipital infarct. This patient had atrial fibrillation, and we believe their cognitive impairment is actually largely due to vascular disease rather than Alzheimer's pathology, because there's no tangles. The mini mental state score is 27, the age is 88, and the MRI shows profound hippocampal atrophy. So we think the explanation for this patient is some incidental amyloid in a patient who really has late, which is a TDP43 related condition, previously called primary hippocampal atrophy and known to be increasingly well recognised in advanced age in people with amnestic dementias. A few final words. Blood biomarkers. Now this is a hot area. Initially the work was all looking at amyloid. This hasn't panned out so well. The hot area now is in the phospho-tau, particularly 181 and now even more so 217. This blood test has excellent discriminating ability between AD and other dementias and between AD and healthy controls. It also has high correlation with tau PET and very good correlation with a positive amyloid PET. Uses a technique called SIMOA, or single molecule array, and there are a number of SIMOA systems in Australia. So ADNeT is working with the ABLE study to validate several SIMOA sites in Australia, and we hope to make this test available to ADNeT-affiliated memory clinics within 2 years as part of a management impact study. So we're trying hard to bring this test to Australia quickly because we think it's going to have a big impact. So finally, AD biomarker summary: PET, amyloid, tau, CSF, and potentially plasma p-tau assays can help determine the underlying pathology of a patient, clarify the diagnosis and prognosis, and direct the patient to the most appropriate therapy and to early intervention clinical trials. So finally, here are those screening centres again, and the email address if you want to refer a patient for the services that we're offering. And with that, I want to thank you for your time.
Thank you very much, Chris, for telling us what we need to know about ADNeT and the screening and the impact that that's going to have on our clinical diagnosis and recruitment. But I guess the fundamental question is, why do we have to be interested in research? Many of us online at the moment are working in memory clinics or maybe in private practice, and we see our job primarily is to form a clinical diagnosis, and we now know from Chris that we can improve our accuracy. We want to obviously establish a management plan, but is research an important part of our role too? The benefits of research into cognitive disorders can be multiple. For instance, we can better characterise the clinical features and the progression if we pool together data from multiple sources, including patients attending memory clinics. We can develop new diagnostic tools and techniques, as Chris has outlined. I mean, all of these tools require patients to become involved in studies, and many of them have to go on and have pathological studies to correlate the imaging with the actual pathology in their brain. We need to develop and can develop better therapies and management, including non-drug options. It's not all about monoclonal antibodies. It's not all about the amyloid hypothesis. There's a lot that we can look at, and in fact, in my own site at the Austin, we are targeting multiple small and large molecules and receptors in our therapeutic trials, not just amyloid and tau. We need to and can give patients and their carers autonomy and the opportunity to be altruistic. And for some, this is a sense of hope. If we tell them about research, they can do something with what some will see otherwise as not a particularly pleasant diagnosis. And I think we need to demonstrate to our fellow clinicians and to the whole community that memory clinics are more than just a one-stop shop for a diagnosis. In fact, it works better if we pool together data, and ADNeT is doing that, of course. And in fact, my own MD thesis, my own postgraduate thesis in 210 was Understanding Dementia, the Role of Multiple Memory Clinic Databases. So we could even go the other way and say, why do we have the right to not inform and direct our patient about their research opportunities? And yet, we know that less than 2% of all patients with a diagnosis of particularly MCI and mild Alzheimer's participate in research trials. We know there's about 40,000 new cases of all dementias per year. That's the incidence in Australia. It works out about 1 case every 6 minutes. And it's even greater if we include people newly diagnosed with mild cognitive impairment. And if we throw in then preclinical that Chris referred to, where there's amyloid but no clinical symptoms, the numbers are even higher. And yet, less than 1,000 are enrolled in formal research trials, even ABLE, with the large database included. So why is this important? Well, let me give you an example of a drug that has been developed and has been trialled in Australian sites. Many of the patients have come from memory clinics, and I think, as Chris intimated, we're going to have their first anti-amyloid therapies which can be administered in these ADNeT memory clinic sites, but let's see how it goes. So it's been recognised that the some of them show slow Alzheimer's progression. Now, blood was taken from these from a site in Zurich, and they found that these people are making amyloid antibodies. In other words, they are turning their Alzheimer's into an autoimmune disease. But unlike most autoimmune diseases, this is good. This is a good disease. One of these autoantibodies was isolated, aducanumab, and it was reverse engineered to become an investigational product that underwent phase 1, phase 2, and phase 3 trials around the world. Now, this is the results from the PRIME study, the phase 2 study, and basically this drug in increasing doses lowered, this is using the SUVR, lowered the amount of amyloid on PET scanning. This is at week 26, and at week 54, we see even further lowering. So this drug can do what we want it to do, remove amyloid from the brain. You can see this also in PET images, where particularly at the higher dose, a highly positive PET scan for amyloid becomes essentially a negative PET scan over a 1 year period. Not only does amyloid become removed, but there's a clinical benefit in the clinical dementia rating sum of boxes. So if we look at, and I'll just move on to the next slide. If we look at aducanumab 10 milligrams per kilogram, that's the data we've got from 6 months to 1 year is this blue line here. We can see that the CDR remains steady, and in fact, compared to the placebo group in yellow, there's a very significant reduction. In other words, a less decline, because decline on the CDR sum of boxes is going up. There's a less decline in those on the highest dose of aducanumab. Now, here's the juice. We've now got the phase 3 trial results. The 2 trials were called Emerge and Engage, and there were about 10 Engage sites around Australia. And as Chris Rowe indicated, the precursor of ADNeT, the brain imaging study in Melbourne, fed patients into the Biogen Engage study. Now, the positive results are actually driven mainly by Emerge, which was mainly United States sites. But when you pool the two, you still get positive results in those on the higher dose. So you've not seen these results before. They've only just come out, and they're really fascinating. So if we look at the CDR sum of boxes again, this is at the week 78. This is the primary endpoint. You'll see a highly statistically significant benefit of the high dose of aducanumab compared to placebo. And we see similar benefits on the MMSE, the ADAS-Cog, and a functional scale, the ADCS-ADL scale for people with mild cognitive impairment. Now, most of these studies include people with mild cognitive impairment due to Alzheimer's or prodromal Alzheimer's, and also include people with early dementia due to Alzheimer's. If we look at the CDR over time, we can see the benefit of the highest dose of aducanumab in purple compared to placebo. And again, this is a statistically significant benefit. It may not seem much, it's about half a point on the CDR, but it is statistically significant. Whether the clinical benefit is convincing to the regulatory authorities, we'll find out in the next few months. Similarly with the MMSE, we see the benefit at 78 weeks. This is the PET amyloid measure through SUVR. Remains steady in the placebo group. You wouldn't expect placebo to remove amyloid, but a significant reduction in the treated group. And likewise, and this is perhaps even more important, well, equally important, we see a reduction in tau in the CSF. Now, we know that this is a drug targeting amyloid, and the Taoists have always said, well, forget about amyloid, it's tau that correlates with neurodegeneration. But lo and behold, if we remove amyloid, we also drive tau levels down. And I think that makes sense because we know basically that amyloid is the trigger, tau is the bullet. If we stop pulling the trigger, then the bullet ain't going to go anywhere. And we see the same also on tau PET scanning in terms of deposition in the medial temporal lobe with the high dose of aducanumab driving this down. Really fascinating results. Now, these patients came from many of the ADNeT precursor sites, the brain imaging study in Melbourne, and many from memory clinics. And this research is still going on. Chris has alluded to the Clarity study, which is a phase 3 study of another monoclonal antibody, BAN2401. Already there's recruitment in several sites around Australia. At my site at the Austin, we've now screened 10 patients and 5 are almost certainly going to go into the study. We're just waiting on some further results. And these people have all had an amyloid scan already before they got into our site because they've gone through ADNeT or they've had amyloid scans done in private. This is the results from the phase 2 study. And again, we see a reduction in SUVR and also the Centerloid value that Chris referred to, particularly in the high dose of this antibody. In fact, this 70 unit reduction is massive in a scale that only goes from 0 to 100. There's other potential molecular targets, the BACE inhibitors and the gamma secretase inhibitors that snip off the A-beta fragment from amyloid have been, the amyloid precursor protein have been looked at. Results are disappointing, but certainly BACE inhibition has not completely died yet. We've also got targets with tau, with mitochondrial dysfunction, and even growth factor type studies, neurogenesis type studies. So the current therapeutic trials that are occurring all around Australia, and this is what Chris said, once they've been screened through ADNeT, we try and match them to a study that they might be interested in and might be suitable for. And there's many options. If they don't get into one study, they might get into another study. Anti-amyloid, anti-tau, we're removing iron, and there's actually a new monoclonal antibody from Roche, which actually binds both amyloid and the brain iron receptor protein. So basically hitting both targets. That's really exciting. We're about to do that study at my site. There's other receptors. There's at least one investigational product being tested in vascular dementia. And we're also not only just looking at products, but looking at ways of measuring their benefits. So we're doing a study of rapid cognitive screening to see if that shows a signal in trials so that we don't have to have long trials with large numbers of people to show a benefit. We need well-characterized patients. Some will come directly from the memory clinic, so you refer them straight from the memory clinic to a trial site, or you refer them from the memory clinic to ADNeT, or you get your referring doctors, your GPs and other clinicians to refer them straight to ADNeT and then on to the trial sites. There's a number of routes. They all work, but we need memory clinics on board. And in fact, if you think about it, if it's going to be expensive or impossible to get good neuroimaging for patients in your memory clinic, of course you'd consider referring them on to ADNeT. The people who are unsuitable, Chris has alluded to, but limited English, very low MMSE, no study partner. If they're outside of a fairly wide age range, multiple comorbidities, particularly if they're neurological comorbidities, and certain prohibited medications. But most trials will take people on a stable dose of a cholinesterase inhibitor. Now, going through ADNeT is one way to get them linked up with a trial site. And most of you who are working in clinics would know your local trial sites. They're often in the same hospital or same facility as the memory clinic. But there's a number of other ways we can find trial sites through Dementia Trials Australia, which used to be called the AC4R. Dementia Australia, used to be called Alzheimer's Australia, also has a research site. So there's many ways you can link a patient up to a site. But if you go through ADNeT, I'll make that easier for you. So I go back to my original question, why research? I'll go the opposite way and say, why would we justify, or how can we justify not offering research to our memory clinic clients and to our private patients that have come to us for an assessment of their cognition? Thank you very much.
Thank you both, Chris and Michael. I think it's been a brilliant start to our webinar series, and I noted that there are 100 participants. So that's really excellent. I think a tribute to the quality of the speakers today. And we have a few questions, and I might start actually. We are a little behind time, but we still have a few minutes to go. So I think we'll address some of these questions. And I might start with a question about memory clinics. And wanted to say that we are trying to bring all these clinics together. And in fact, we will list the clinics on our ADNeT website and also develop a tool, widget, so that one can search for the most appropriate memory clinic. Now, that does include, and this is one of the questions, private clinics. And certainly it's not exclusive to public sector clinics. The way we look at memory clinics is that usually these are multidisciplinary clinics. But certainly there are clinics that are run by solo practitioners, and we actually want to capture them as well to see whether there could be methods by which we could enhance these clinics. And there is a lot being planned to actually support these clinics, develop assessment tools, harmonized tools, develop a norming tool for them, also looking at various post-diagnostic measures as well. So in fact, you can go on to our ADNeT website and follow the various activities that are happening in the clinics as well. And if you are interested in being part of this network, please get in touch with us and we will leave an email contact, or alternatively, you can certainly go to the ADNeT website and send us contact information. So with that, I think I'll come to some of the questions that are being asked. One is, of course, with Step Up, and maybe Chris, you want to address that, because we have this volunteer register being prepared for ADNeT, and then Step Up, Yun-Hee Jeong, Professor Yun-Hee Jeong from Sydney University has already developed Step Up, and we can probably talk briefly about the collaboration between the two groups.
Yeah. Yes, so we are a supporter of Step Up, and we think that's been a great development. Our portal is a little bit more tailored, and it includes, for example, the ApoE4 testing, and I think we're going to work together. We hope that actually we will be recruiting through Step Up and basically mutually supporting each other. So certainly Step Up's done a good job. It's got a pretty good profile. It was initially developed just in Sydney and WA, but it is now spreading out more nationally, and we certainly do intend to continue and probably grow our support for Step Up.
Okay, thanks very much. And maybe Chris, while you're there, the two questions in relation to amyloid and tau. One is, of course, with p-tau. It's a technical question. What about frozen samples? Do you think p-tau is sufficiently stable to be sort of measured in frozen plasma samples?
Yes, it does appear to be quite a robust test, and there's no doubt you can ship your blood to the other side of the world and get reliable results.
These are archival samples, say, if they've been stored for a few years. Do you think that's still okay?
I'm not an absolute expert, but if they've been frozen for all that time, I think they'd still be okay.
Okay. The other question, I guess, asked is, why this focus on amyloid, really? Is this, does it subscribe to the amyloid hypothesis of Alzheimer's disease? And maybe a short answer to that.
I think the evidence is quite compelling that amyloid is the earliest detectable change. There's a bit of neuroinflammation, probably when amyloid starts appearing. And then once it's got to a certain level, you see the tau explode throughout the brain. So it's then it's the tau that correlates with the damage, with the atrophy and the cognitive impairment. So it does have some implications. It may be too late to treat amyloid in somebody with established dementia. You may be better off targeting the tau. But this still does help, I think, push towards the need for early intervention with anti-amyloid therapies.
If I could make a point there too, I mean, I think some of the skepticism about amyloid has been because certainly you can find amyloid in the brains of people who are cognitively normal, as Chris indicated. But my response to that is that we're actually just picking people who haven't yet had time to develop the clinical features. In other words, they've died with their amyloid before the amyloid has led to the syndrome of MCI or dementia. And secondly, we do know there are some conditions that mimic Alzheimer's disease, such as late. In fact, those of you who are enrolling in Simon Lewis's masterclass, Diseases of the Aging Brain, will hear a talk about late in early October. And Chris alluded to that in his talk. So there are conditions that look like Alzheimer's but are not Alzheimer's, and that led to the idea, well, maybe amyloid isn't the cause of, isn't the one, the culprit, if you like. But increasingly, we're recognising that the vast majority of people who die with a clinical syndrome that looks like Alzheimer's have had that because of amyloid and then tau.
So I think also, thanks very much, Michael. That's very helpful. Just in terms of, this is Lyndon Newton's question. So the 30% who did not have amyloid were misdiagnosed. You agree? They did not have Alzheimer's disease because by definition, you do not diagnose Alzheimer's disease if someone does not have amyloid in the brain. That's, would you accept that, isn't it? Yeah. That's generally accepted now.
Yeah, that's what we used to call SNAP, suspected non-amyloid pathology, and a lot of that is turning out to be TDP-43 or late.
Sure. Okay, Michael, here there's another question for you. So the question is from Raju George. Says, I work in rural New South Wales, and most patients decline to travel to metropolitan area for participation in studies. This is especially true in the current scenario. What can we offer such patients? I have a few young CSA positive MCI, Alzheimer's MCI patients who decline to travel.
Well, we've been fortunate in Victoria. We've had COVID, and COVID has taught us a different way to do research. So, for instance, in our graduate study, which is the Roche monoclonal antibody, gantenerumab, we've got 37 participants at our site, and a substantial proportion of them, particularly from more distant parts of the state and even some from interstate, have elected to have the drug given at home. So we have home nurses go and administer the drug. More and more of the endpoints, the psychometric endpoints, can be done via telemedicine, telehealth. People still do have to come in at some stage for safety checks, including MRIs, but we can vastly reduce the amount of time that people have to come physically to our site. And I remember Lon Schneider about 10 years ago talking about the Apple virtual trial site, using very innovative endpoints such as sensory sensation detectors in the house, motion detectors in the house, and all sorts of things that can be used as endpoints in various studies where a patient doesn't even have to leave their own home. So no, I don't think rurality completely negates involvement in a clinical trial. I don't think we're going to get PET centres outside of the capital cities, but Chris would be a better person to speak on that.
So,
there actually
some
are
advice, Chris, and maybe you can also answer this. If there's some advice, if someone from a rural area does want to participate, who do they contact?
Well, they can go through ADNeT once there's an ADNeT in the capital city, and they can do the initial screening. They will have to come into the capital city to go to ADNeT. But as I said, most of the subsequent trial participation doesn't necessarily have to be that way. Now, I know there are mobile memory clinics. There's one based at the Hunter that travels around the region there and basically does the memory clinic from the back of a van. So they can achieve clinical diagnosis that way. But the neuroimaging, which 100% of my patients will have some neuroimaging, usually MRI and amyloid PET. The neuroimaging, they will probably have to come to a rural site. But Chris, you were going to say something about PETs in a station wagon or something.
Most major regional centres have a PET camera because of its extensive use for oncology. You'll find PET scanners in Bundaberg, Darwin, Lismore, Bendigo, Ballarat, etc. They are out there in the major regional centres. What's missing really is the key clinical driver for doing trials in those areas, and perhaps a sufficient number of suitable patients to make a trial site out there viable.
Sure, thanks. Thanks very much. And I think the other question, and maybe this could be the last question we take, is requirement of English, really, and if English is a second language, really. I think that's something that perhaps trials ought to look at. I mean, I can see the reasons why, but certainly I think this is something that, I don't know, Michael, whether you have any comment on that.
Oh yeah, look, I'm quite concerned. I mean, it is a form of discrimination. I absolutely agree with that. I've had people speaking Cantonese or Mandarin who are prepared to fly to sites in either Hong Kong or Singapore to become involved in a trial after I made the diagnosis. And interestingly, I contacted those sites and they say, no, we only take English-speaking people here too, which is fascinating. But there are trials that use local languages around the world, Spanish, French, etc. But here in Australia, nearly all of our investigational products for Alzheimer's require cognitive endpoints, which can only at this stage be administered to people who are English speaking. It is unfortunate, I understand that, but that's the way it is. Maybe when we use computerized cognitive testing, and we're testing one of those platforms now that Roche is supporting, we might be able to move away from English speaking. Maybe.
Well, I think with that, thank you again. Thank you to our speakers. for the excellent presentations and for you all, to you all for attending our first webinar and making it such an exciting event. And we look forward to seeing you again, and also your active participation in Memory Clinic Network. In fact, that's really the most important aspect of this. Thank you and bye. Take care.